and hello everyone. Thank you for joining us at this webinar. We are seeing remarkable new trends in drug discovery, and I believe that RNA-targeted drug discovery is one of the most promising frontiers in small molecule therapeutics right now. Today, I want to give you a practical picture of how we are approaching it at Veritas In Silico. As you know, most small molecule programs have focused on proteins. The tools, the assays, and the medicinal chemistry intuition are all well established for protein targets. But proteins are only part of the story. The human genome encodes roughly 20,000 proteins, but the transcriptome contains over 500,000 RNA molecules. To make any protein, the cell must first transcribe the gene into RNA. And beyond that, RNA molecules play direct, essential roles in disease. Some control how genes are expressed. Others are critical to the life cycle of viruses. So here's the question that drives our work. What if we target RNA directly with small molecules, just as we target proteins today? In 2020, risdiplam became the first approved small molecule that works by directly changing how a human RNA is processed for spinal muscular atrophy. That approval was a landmark. It showed that RNA can be a genuine drug target. Since then, many groups in academia and industry have started working on RNA targets. Untranslated regions, splicing signals, repeat expansion sequences, and many more. The field has grown considerably, and there is strong momentum. But the field still faces real challenges. Today, I want to be open about those challenges and show you how we are addressing them through our platform, aibVIS. I will walk through two parts of our recent work. First, a screening workflow that uses AI to find active compounds more efficiently. And second, a structure-guided optimization workflow which combines molecular dynamics simulations with quantum chemistry to understand why compounds bind to RNA and how to optimize them. Before I dive into our recent work, let me take a moment to talk about the challenges associated with RNA-targeted drug discovery, because they explain every design decision we made in building aibVIS. The first challenge is flexibility. Proteins usually fold into stable three-dimensional shapes with well-defined binding pockets. RNA, on the other hand, does not behave this way. The same RNA sequence can adopt different shapes depending on temperature, salt concentration, or the presence of nearby molecules. This makes it hard to define clear, stable binding site and hard to predict how a small molecule will interact with it. The second challenge is limited structural data. Decades of X-ray crystallography and NMR have produced a large library of high-resolution protein structures. For RNA, that library is much smaller. Many important RNA targets have no experimental structure at all, which limits the starting point for computational design. Third, structure-activity relationships are often unpredictable. A small change in a compound can cause an unexpected change in activity, and the reason is often not clear from the structure alone. Without that understanding, synthetic effort can easily go in the wrong direction. Finally, finding true binders is experimentally difficult. Many compounds bind RNA nonspecifically, and distinguishing those promiscuous binders from genuine selective binders requires orthogonal assays. On top of that, most existing RNA binding assays cannot be scaled up to the compound numbers needed in early drug discovery. So these are the problems that motivated us to build aibVIS, not as a single solution, but as a set of connected tools designed to support the full early drug discovery process for RNA-targeted small molecules. From identifying a target, to finding active compounds, to understanding them and optimizing them. The first stage is target identification. We use computational methods to analyze RNA sequences and identify regions that might form stable pocket-like structures, the kind of shapes that small molecules could bind to. The second stage is screening. We test compound libraries against RNA targets using a fluorescence-based method called QFRET, which measures whether a compound stabilizes the RNA structure. I will explain QFRET in detail shortly. The third stage is hit to lead. Once we have initial actives, we used cell-based assays and a range of biophysical methods, including BLI, ITC, and NMR, to confirm binding, understand potency, and begin building structure-activity relationships. The fourth stage is lead optimization. We use X-ray crystallography and NMR to determine the three-dimensional structures of RNA compound complexes. Combined with molecular dynamics simulations and quantum chemical calculations, this gives us the mechanistic understanding needed to optimize compounds in a rational way. So together, these four stages form a continuously connected system, each one informed by the others. As I mentioned earlier, today we will focus on screening and lead optimization. Both start from the same experimental foundation, QFRET. Here is how QFRET works. We attach two labels to an RNA probe, a fluorescent dye and a quencher. When the RNA is folded, these labels are held together and fluorescence is low. As we heat the sample, the RNA unfolds, the labels separate, and fluorescence rises. The temperature at which the RNA is halfway between the folded and unfolded is called the melting temperature, or TM. When a compound binds and stabilizes the RNA, it takes more heat to unfold it, so the TM shifts upward. We call that shift delta TM. A larger delta TM means stronger stabilization. With that foundation in place, let me explain how we built a smarter screening workflow on top of it. We built an AI-augmented iterative screening workflow called AISLAR. We start by screening a small subset of the library, use machine learning to predict activity across the rest, then select the next batch, screen again, analyze the data, and feed the results back into the model. With each cycle, the search becomes more focused. what was once a one-way process becomes a learning loop powered by AI and automation. And this method is not just theoretical. Similar iterative methods have already shown strong efficiency gains in protein-targeted programs, giving us the confidence that this concept could be adapted for RNA. We validated AISLAR on an in-house dataset of about 70,000 compounds screened by QFRET against 2 RNA motifs within the p53 messenger RNA. Just so you're aware, reducing p53 expression is a promising strategy to prevent acute kidney injury. Motif 1 is a structured element in the coding region of p53. Motif 2 is located within the p53 iris, an internal ribosome entry site, which is basically a structural element that controls how the p53 protein is made. Because motif 2 is believed to play a regulatory role in p53 expression, it represents an interesting RNA target for small molecule modulation. We labeled the active compounds, trained separate models for each motif, and ran iterative screening simulations to measure how efficiently AISLAR could recover active compounds compared to random selection. For both motifs, AISLAR recovered over 70% of all the active compounds after testing only about a third of the total library. To put that in practical terms, you can identify the large majority of useful compounds while running only a fraction of the experiments you would otherwise need. For an organization with limited screening capacity or working with expensive RNA assays, that efficiency gain has a direct impact on what is feasible within a given project timeline and budget. Compared to random selection, this shows clear and consistent enrichment across both targets. From here, I will focus on motif 2, where we explored the system in more detail. When we mapped the motif 2 actives in chemical space using a dimensionality reduction method that places chemically similar compounds near each other, we found that the recovered compounds are not randomly distributed. Instead, they organize into distinct chemical clusters. This is important because it suggests that the model is learning meaningful patterns associated with RNA recognition rather than enriching isolated active compounds with no clear chemical relationship. Next, we examined clusters recovered by AISLAR. Cluster 1 shows strong stabilization. Cluster 2 is more moderate, and cluster 3 shows little to no stabilization and was not recovered by AISLAR. Clusters 1 and 2 are particularly valuable because they give us chemically related compounds with interpretable differences in activity, exactly the kind of series that supports rational optimization. Looking more closely at cluster 2, we identified a recurring pharmacophore, a basic group linked to a thiophenyl-quinazoline core. This shared scaffold gave us a starting point for structure-activity analysis and rational optimization. However, computational analysis highlighted a potential safety concern. These compounds were predicted to block hERG, a cardiac ion channel commonly flagged for basic compounds in early medicinal chemistry. So we designed an analog that preserved the key pharmacophore geometry while reducing overall molecular basicity. The resulting compound retained RNA stabilization activity and was predicted to be a hERG non-blocker. This is an important aspect of AISLAR. It not only identifies active compounds, but also finds chemically interpretable series early enough to support rational optimization, including optimizing for safety. To understand the structural basis of binding, we turn to X-ray crystallography. We determined the crystal structure of the p53 iris internal loop. As some of you may know, RNA crystallography is technically challenging because RNA molecules are often difficult to pack into well-ordered crystals. To address this, we engineered a construct optimized to improve crystal packing and diffraction quality. The resulting structure provided a three-dimensional framework for subsequent modeling studies. However, as I mentioned earlier, RNA is highly flexible, and a crystal structure represents only a static snapshot of the system. So, to understand how the compounds behave within the dynamic RNA environment, we combined docking with molecular dynamics simulations. We ran multiple independent simulations for each compound, allowing us to sample different regions of conformational space. Although the exact poses vary between simulations, binding consistently localizes to the same pocket. The core of the compound is between two adenines in the binding pocket, stacking face to face like a filling in a sandwich. This type of interaction is known as pi-pi stacking and is commonly observed in small molecule recognition of RNA. An adjacent group also forms a hydrogen bond with the phosphate backbone of another adenine, and additional contacts further stabilize the complex. This structural picture matches the pharmacophore AISLAR had identified from purely experimental data. An aromatic core for stacking paired with a basic group engaging the backbone. In other words, two independent analyses, one data-driven and one structure-based, converged on the same answer. That convergence gives us genuine confidence that we are capturing something real about how the compounds recognize this RNA target. If you'd like to explore this research further, the full details are now in our recent paper in Small Science. Okay, so far I've shown how AISLAR can efficiently identify and characterize RNA binding compounds. The next challenge is turning those hits into optimized leads. And for that, I will switch to a structure-guided approach that combines three-dimensional structural data with a method known as fragment molecular orbital, or FMO. Together, they form a design, make, test, analyze cycle for rational optimization. To develop the workflow, we worked with a well-characterized model system. A stem loop RNA with a bulged-out guanine called A4G, and a fluoroquinolone derivative called KG022. We identified binders by QFRET validated them with BLI, ITC, and NMR, and designed analogs based on the solution structure of the A4G KG022 complex. This gave us a direct, well-characterized path from screening to structure and to rational optimization. We made several structure-guided modifications to KG022. Based on what we could see in the structural model, we expected improved binding. However, when we tested the early analogs, the improvements were modest, suggesting that the structural model alone was not sufficient to guide analog design reliably. And that's what led us to FMO. FMO divides a biomolecule into fragments and calculates interaction energies between them. This tells us which residues interact with the compound and what drives binding affinity. In principle, FMO provides a powerful framework for rational optimization. For example, if binding is driven mainly by stacking at a particular nucleotide, we can design compounds specifically to strengthen that interaction. When we plotted the interaction energies from FMO against the measured binding energies from our ITC experiments, we found that there was a strong correlation between the two indices and predicted that two new analogs would outperform the parent compound. But when we tested them, the predictions did not hold. We hypothesized that the problem was RNA flexibility. Our FMO calculations had just used a single static NMR structure. But RNA, as I've said earlier, is dynamic, and a single snapshot misses much of the conformational landscape that is relevant to binding. The binding energy the ligand actually experiences reflects an average across many RNA conformations, not just one. To address this, we combined FMO with molecular dynamics, MD-FMO. We dock the compounds into the RNA structure, run molecular dynamics simulations to let the complex explore a range of conformations, cluster the resulting trajectories to identify representative structural states, and then apply FMO calculations to structures from each cluster. MD captures the conformational landscape, while FMO evaluates the interaction energies within it. The key shift is that instead of asking, how does this compound interact with this one structure, we are now asking, how does this compound interact across the full range of shapes this RNA can adopt? And the improvement was substantial. Across our expanded set of analogs, the interaction energies from MD-FMO now quantitatively match measured binding enthalpies, confirming that accounting for RNA dynamics is essential for our accurate prediction. FMO also gives us something that binding experiments alone cannot: a residue level map of where the interaction energies come from. We can identify which specific nucleotides contribute most to binding and through what type of interaction. The highlighted residues form strong stacking and electrostatic interactions with the small molecule, especially around the bulged out guanine. This type of detailed mechanistic analysis not only explains the observed binding affinities, it also points directly to where future analog design should focus in to improve potency in a more systematic way. So to wrap up, we focused on two challenges: improving the efficiency of RNA-targeted screening and guiding hit-to-lead optimization with structural insight. AISLAR provides an AI-driven framework for predictive screening, while MDFMO enables quantitative and mechanistic understanding of RNA small molecule interactions. Together, these approaches form part of our broader effort to build an integrated computational and experimental pipeline for RNA-targeted drug discovery. RNA-targeted drug discovery is still at an early age compared with protein drug discovery, and difficult challenges remain. But we now have a combination of experimental methods, structural biology, and computational tools needed to study RNA systematically rather than relying purely on trial and error. So looking ahead, we are expanding our use of AI to drive RNA-targeted drug discovery further. First, we are exploring deep learning to predict RNA structures for faster and more reliable target identification. Second, we are developing next-generation screening models trained on our growing data sets across multiple RNA targets to reduce false positives and improve hit prioritization. Third, we are building machine learning tools for predicting binding affinity and drug-like properties. And fourth, we are working toward SAR modeling and PK/PD predictions to guide and de-risk in vivo studies. These advancements aim to make RNA-targeted drug discovery more precise and efficient, a future I think we're all eager to see unfold. Before I end, I want to take a moment to thank the incredible team at Veritas In Silico who made this work possible, our academic collaborators for their expertise, and our industry partners for their collaboration. We've taken on a challenge that is truly monumental, but one that matters deeply. As Shoji-san said earlier, at the end of the day, this work is about improving the lives of patients. There is still a lot to be done, and we can't do it alone. So if any of what you heard today resonates with you, we'd like to talk. Thank you all for your attention, and I'm happy to take any questions.